Запись сейчас пойдет.
Good morning, dear colleagues. Today's theme is development of the heart. Extremely difficult and extremely important theme. Let's begin from the most early stages. Stage of formation of the primitive heart tube. What is this? It's written that it's 20 days of embryonic development. We see now the embryonic disc, which is located superiorly to the yolk sac, and in this period embryon is quite flat, three-layered disc. Now we look at this disc from the posterior view, or we can say superior view. On this stage, you can see that there is line, midline, which divides the embryo on two symmetrical sides, left and right. And we can declare that from this period, from 20 days of intrauterine period, the human person has bilateral symmetry. From this moment, from 20th day of intrauterine period. On this 20th day, we can see several small details which later should be very important for understanding what it's happened. Now, we have cutted the embryonic disc in mid-sagittal plane, and here from right side of our section we can see how it looks like. So I promised that there is three-layered disc. Really, this blue color is color of ectoderm. This green color is color of cord, primitive cord, and this yellow color belongs to yolk sac, and it will be entoderm. So ectoderm, cord, and yolk sac. And on these 20 days, we can see that ventrally is placed future heart, which is connected with two tubes, one coming from posterior part and another making outflow to anterior direction. Let's continue. we can see another stage of development. This yellow endoderm begin to be so named foregut. From it, in future, the pharynx and esophagus will develop. And again, very interesting thing that future heart is laid practically on the neck region of embryo. This is future head with brain. Its cranial portion of ectoderm will form the brain, and heart is located near to the head, practically on the neck region. And what can we see now? That the gut, the future intestine, practically is formed, and the future heart is here. Inside of it, we can see red color tube, endocardial tube, which is part of future heart. And around of it, we can see structures which were developed from mesoderm, third layer of embryo. So, heart is developing from mesoderm and from two different sources: endocardial tube and the epimyocardial plate. They are, in the first stage, paired structures, that there is left and right structures, quite symmetrical each other, and then it will be fused to one structure. So let's try to understand. This is developing forebrain. This is foregut, primitive heart, and close to it there is so named septum transversum, which is divided heart from anterior part of the embryo. It is interestingly that we can find just now at 20th day so named oropharyngeal membrane. Oropharyngeal membrane. This membrane is situated on the place of isthmus fautium of the adult man. Do you remember isthmus fautium? It is place where oral cavity connects with pharynx. So we can say that oral cavity is lined with blue color line. Why? Because it is from ectoderm. And from posterior view, oropharyngeal membrane is lined with this yellow line. It means it is developed from entoderm. And when this oropharyngeal membrane will be dissolved, so the outer environment could enter the internal environment, and our primitive mouth shall connect with foregut, the future pharynx and esophagus. But what about heart? For understanding, we shall look at developing heart from transverse plane. Let us make such kind section and try to see en face, as French says. So we can have such kind diagram. Let us try to find all structures. This green is chord or notochord, which take place in forming of the spinal skeleton and skeleton of the head, the skull. This is delineated with blue line, is from ectoderm developing the neural folds and neural groove. And here, delineated with yellow line, is forming foregut. And in this moment, we can see that there are two endothelial lined hard tubes, right and left. You can see that this process is very rapid. So in several hours, these two endothelial lined tubes becomes closer to each other and then fused with each other. In the same period, the neural groove becomes neural tube, and from this tube will develop in future the spinal cord, and in cranial pole of the embryo, the brain. Now we can see that foregut, the primitive intestine, is closed and it's lined with yellow line. It means it was developed from entoderm. And also we can see the secondary cavity of the organism, the pleura pericardial cavity. Or we can say that this is cellome. Cellome is secondary cavity of the embryo. And now, how it develops. Oh, it's very interesting. Now we can see the single tube. Two paired tubes become fused and a single tube appears. This is first stage of tubular heart, stage of simple tubular heart. What is the main function of heart? It's pulp, which necessary for moving blood. And blood is necessary to provide nourishment of each cells of organism. When embryo was quite small, less than 1 millimeter, it was enough diffusion for nourishing each cell. But at this stage, the length of embryo becomes about 2 millimeters, and diffusion isn't enough for nourishing each cell. So appears vessels which serves as transport system for nourishing structures. Let us try to understand each detail. So, we can see that primitive heart tube is surrounded with part of pericardial cavity, and in this stage it has dorsal mesocardium. This term, you understand, it's similar to mesoovarium, mesenterium, mesocolon, mesoappendix. It means double layered structure which hold the organ to the dorsal surface of the secondary cavity. And also heart in this period has dorsal mesocardium. And the surface of the future heart is delineated with epicardium, and this pink structure is myocardium. So in this period, primitive heart tube consists of 3 layers, which is the same like in adult person. Endocard, myocard, and epicard. On the dorsal surface of the organism, from both sides of notochord with green line, with green color, we can find 2 large vessels which are absent in adult men. They called dorsal aortas. So there are 2 dorsal aortas in this period. This is narrow tube, and this is somit. Somit is paired structure, that's why you can see right and left somit. And what will be later? Later we shall see, like heart, this tubular heart, while developing, make some folds inside of pericardium. Now here it is, this heart tube, the primitive heart. From the early stage, heart contracts. Contraction is quite rapid, from 100 to 180 contractions per minute. And around of heart tube we can see this sac, pericardial sac or pericardial cavity, and heart develops very rapidly inside of this pericardial sac, pericardial cavity. So this is 22nd day. From dorsal part of the embryo we can see only neural groove. There is no neural tube, but nevertheless heart already consists. Heart already contracts. In this stage we can divide this tube artificially on left ventriculum and right ventriculum, but they are part of the same tube, but left ventricle is more posteriorly located and right ventricle is more anteriorly located, and outflow region of the primitive heart is aortic sac. It means that blood goes out of heart tube through this aortic sac. It's good for us to remember what is arteria and what is vein. Arteria is a vessel which is going out from the heart, and veins are the vessels which goes toward the heart, which carries the blood to the heart. Also, I'd like to demonstrate here this yellow part of the scheme of the diagram, which is yolk sac. Yolk sac consists in this period, and from it the foregut is forming. And from this yolk sac, several umbilical vessels are beginning, and they enter it to the heart. Now, let us see which way will develop the heart in next several days. Be attentive. 23rd day, 24th day. The heart is developing. It becomes longer, and that's why it's impossible to be inside of this pericardial cavity without any looping. So heart tube forms this loop. 26th day, the day of looping. First looping of the heart tube. And then you can see it isn't enough place for developing heart in this place, in this cavity, and that's why it becomes another way arranged inside of pericardial cavity. Before to look another view of this diagram, let's find the most important structures. So this is pericardium, and inside of it this heart loop, it means tube, becomes very folded and it fulfills the whole cavity and several times looping inside, and from posterior surface we can see here the closing neural crest, which becoming neural tube. Several hours later, it becomes closed tube. And now we look at this from sagittal view. This is from transverse section, and this is from sagittal view. You can see that heart is twice looping. This is inferiorly now located venous part of adjacent vessels, and superiorly is located outflow part of heart and outgoing vessels, that means arteries. So these are veins and this is artery. And in this view we can again to see at the formation of heart tube to understand in detail all phases of transformation. Again, these are the veins and these are the arteries. 23rd day. Becomes greater, and what can we find in this heart? Most posterior part is common cardiac vein, and then more anteriorly from backside there is atrium, common atrium, and we can say common ventricle, but from the first stages of looping, this single ventricle is divided to these two parts, primitive left ventricle and primitive right ventricle. If we shall cut it transversely, we can find that they have the single cavity. There is no division between primitive right ventricle, primitive left ventricle. But nevertheless, we can propose that this line will be future border between future septum between left and right ventricle. So we can see that next day it becomes larger, and we can see it from two positions, ventral view and lateral view. On both positions we can see the loop which forms heart tube in this period. All veins is collecting to this sinus venosus, those part which direct the flow of blood to primordial atrium. So we see that atrium in this stage is from backside and ventricles are from anterior side. Next lesson, when we shall use cadaveric specimen while teaching, we can find that atrium is impossible to demonstrate from anterior view. It's necessary to turn the specimen with posterior surface to your face, and only in this situation you can find atria. I'd like to say that from 24th day atria are located from posterior side of the heart. It's impossible to demonstrate them from anteriorly. Only two special parts of atria which called auricula. Auricula dextra and auricula sinistra. We shall see them later. What can we see now? We can see that atria moves superiorly, not only in posterior direction, but also superiorly. And that's why appears these two rounded shape structures which embrace the future aort and now outflow region from right and left side. These are the auriculae, auricula dextra and auricula sinistra. it would be good if you make photo from this structure, because it's very interesting on next lesson to compare this structure of 24th day with adult heart and to find that right atrium is here, left atrium is here, right auricula is here, left auricula is here, but both atria are located from posterior surface. It's impossible to demonstrate them anteriorly. And then we shall find the contributions of secondary heart field for development of heart. So this is ventral view, again narrow fold, the same blue color, the mesoderm of red color, and after moving sagittally, these paired primitive parts of the heart fused with each other. And very important stage from lateral view, which we can see now. Let's try to distinguish all the detail. This is future brain. This is so named branchial arches. Inside of them, fifth cranial nerve develops, and this is developing heart. So from lateral view we can distinguish that developing heart is located quite close to developing head. So one type of heart dystrophy is cervical dystrophy. It's written that this is primitive right ventricle of red color and of green color, this outflow region, which in future becomes aorta and truncus pulmonalis. And now the same from anterior position. You can distinguish two ventricles, right and left, two auriculas, right auricular and left, and atria are not seen, because they are placed posteriorly to these structures. It's necessary to turn this scheme, this specimen, from the backside. And also it's interestingly that there is only one vessel which outgoing from the heart to the cranial direction. Let us call all the structures again. Here are the designations. And now let's try to understand what it will be if we cut longitudinally our heart tube. Let us make the division, the cutting. So we have made the cut in longitudinal view of the heart tube. We can see only one loop now. This is loop, and this is region of atrium, ventricle, and this is outflow region. And there are several arteries which develops in forming bronchial arches. So these vessels are externally according to the heart. So let's again try to understand all the designations. So if it's the atrium and this is the ventricle, so between them appears the narrowing part, so named atrioventricular canal, very narrow atrioventricular canal. For it appears is necessary that from both sides externally there were a lot of special cells which provide this process of growth. So let's look at this region and try to understand that there is migration of special cells and tissues from myocardium into endocardial directions. From myocardium, cells migrate to the direction of endocardium, and from endocardium a lot of mesenchymal cells migrate in this region. So around of the atrioventricular canal we can see now the special tissue which provides narrowing of atrioventricular canal. And now, next very important question, realignment of primitive heart tube. So, due to this process, we can see inside our heart is cutted. We can see inside two curvature, inner curvature and outer curvature. And these curvatures are in forming common ventricle, but now it's written that this is right ventricle. Yeah, but there is no division of ventricle into right and left side by any septs. And there is opening to the atrioventricular canal. This opening from those chamber which is out of this surface this diagram. It's posterior to our screen on which you see this picture. So atria are from those side. They are not seen here. This is only auriculars, the parts of atria. So the atria are posteriorly to the scheme, and the connections between atria and ventricle is designated as opening to the atrioventricular canal. In this stage, the myocardium becomes thick, and you can see the wall of the heart is quite developed. Again, let's designate all the structures and try to see on this square with process magnification. So we can find that migrating cells form so-named cushion. These cushions are doubled. This is from cushions which surrounds the outflow region, and this is cushion, endocardial cushion, which make the narrowing atrioventricular canal. So we can see primitive atrium and primitive ventricles, and between them there is very narrow space. The process of migration is designated here. So again, we can see the directions of these migrating cells. So due to this process, the shape of the heart changes. You can see the migrations of the outflow region, and from this point of time there is very important situation which we should understand in all periods of postnatal life. Please be attentive. This is outflow region, and in this moment the atrioventricular canal divided into left and right. So in this moment, practically there is a common ventricle. The separation of left ventricle from right is absent. But there are two atria at the moment, right atrium and left atrium. From those atria, the blood enters to the outflow region. And you can see that blood flow from left and right atrioventricular canals, they cross each other. It's very important because it is the first step to make the crossing of future aorta and future truncus pulmonalis. Truncus pulmonalis and aorta in adult men is crossing spirally, one according to other. So you can see it's very complicated development, but we should know it to understand all the cardiac failures which take place before and under the birth, and to know the way of the development, we understand the development of the heart. So, next part of our lecture we shall make 1 week later. Now I ask the leaders of groups to make the marks in our electronic bearers system who is present. Thank you. All another are free.